work. So I think we will roll is in it. I think we gave more time. The 3-minute thesis doesn't have any questions. So it's going to go quite bang, bang, bang. And yeah, we'll be fine.
Yeah, Abigail, do you want us like to be kind of like a stern for time? Do we want to remind people?
3 minutes are 3 minutes.
Yeah, yeah.
Okay.
And absolutely 3 minutes. That's
Speaking of time,
yeah,
it's 1:30.
Yeah,
Shall we get going?
1:30. Okay. We didn't, most people came back.
That's right.
Will go. Joanne, you're welcome.
All right. Well, so I'm here to talk about some of the work that we've been doing, developing rapid tests to improve surveillance of mosquito-borne diseases. A number of you will know that I have been developing rapid testing strategies. So just a little bit of background for those of you that don't know. We've developed a really quick way to sensitively detect diseases by looking for their DNA. But instead of having to take the samples back to a lab, we believe that this could be just done right there where you take the samples, because it's a simple 5-minute room temperature, single step, add the sample into a liquid that we made, mush, mush, mush with a homogenizer, and then after 5 minutes, take it out, pop it in a little tube where DNA is amplified really quickly, 5 to 10 minutes at a single temperature, 39 degrees. And then take some of that out, pop it onto one of these lateral flow strips, otherwise well known as a pregnancy test strip. And then 5 minutes later, you can see a result by the appearance of a line here at the test. If only one line appears at the top where the control is, that means that there was enough liquid added to the strip for things to wick up and the control worked, but what you were looking for isn't present. And we've also done some work developing multiplex tests, which I'm not going to go into very much today. We've published various aspects of all of these technologies except the extraction, which is new, so that hasn't been published. And if you're interested in the publications, I'll put them up there. If we're going to compare this to what is normally happens, so normally people take the sample, then they come back to a lab and they have to process the samples to get the DNA out, or the RNA out if it's an RNA virus. So that's a 45-minute plus procedure that either uses a robot or a column and a centrifuge, fairly bulky lab equipment that just isn't well suited for point of care kind of testing. And then after that, most people would normally do a real-time PCR, which is a machine that looks like this. This is the Agilent one, that takes 2 hours or more usually to then get a result that a computer has to read. So we've taken essentially a 3-hour procedure and got it down to about 20 minutes without needing any of these amazing pieces of equipment. So we're quite excited. We've been working on lots and lots of different tests, but today we're going to focus in on our mosquito testing. And in particular, because I only have 10 minutes, we're going to talk about dengue specifically. This work was mostly done by Madija, one of the PhD students in our group, who would be presenting today, except that she's about to have a baby any minute. So well wishes to Madija if you're listening in. I hope that everything goes really, really well. There are about 390 million dengue infections each year. And importantly, there are actually 4 different types of dengue. And if you happen to get dengue, a different type to the first one you had, you're at risk of very, very severe disease and even death through hemorrhagic fever and shock. And so there's lots of tests for dengue, but there aren't very many dengue typing tests, particularly rapid ones that could be used to type mosquitoes so that a community could check and see if another dengue that they haven't had before has entered into their community and make sure they wipe it out so that people don't get these terrible diseases. So Madija has gone and made a test for each one of these different 4 types of dengue. So this is just her dengue 1 test, but she's done this and we get the same results for all of them. If we take a synthetic RNA molecule that we know its copy number, that's got the sequence of what we're trying to detect, and dilute it down, We can detect down to about 400 copies. You can see a little faint line there. When we repeat this a few times, in fact, only 75% of our testing comes up positive at that, but 100% come up positive at about 1,000 copies. And that's a pretty good sensitivity for trying to detect dengue in a mosquito, because when the mosquito gets infected with dengue, it becomes a systemic infection. The virus actually grows in the mosquito, and then it secretes it in its saliva when it bites you to suck your blood. And so detecting down to about 4,000 copies should be enough to detect a systemically infected mosquito. And we've done that thanks to mosquitoes that were given to us by Leon Hugo at QIMR and also Andrew Vandenherk at Queensland Health Forensic and Scientific Services. So if we take uninfected mosquitoes, and we've done this with 15 mosquitoes in this experiment, none of them showed that little test line, whereas the mosquitoes that were infected with dengue 1, the 7 mosquitoes that we had, all of them came up with a positive test line. So that's really promising. Interestingly, there's another bacteria that infects mosquitoes called Wolbachia. And if the mosquitoes that normally give you dengue happen to be infected with Wolbachia, then it actually prevents the mosquitoes from transmitting dengue. And there's a big controlled release program going around the world that's run by the World Mosquito Program, where they're actually taking mosquitoes, infecting them with Wolbachia, and then releasing them into local populations so that when they breed, the Wolbachia spreads. And they're getting some very, very interesting results showing the drop in dengue. And this has also been going on in Australia in Cairns, and we're now dengue-free, which is really exciting. So we were funded by the Bill and Melinda Gates Foundation to develop a test for Wolbachia so people could check where the Wolbachia was going. So that one was really nice and sensitive. We could detect down to 2 to 20 copies per microliter. If we used a synthetic DNA to test. And now we've done a number of different experiments, but this is the one that I like the most. And that is we took freshly infected mosquitoes and uninfected, and showed that all the infected ones were positive on our Wolbachia test and the uninfected ones weren't. We then took mosquitoes that were just left in a trap for a week. They were all shriveled up and dried, and didn't really expect them to be positive, but the 3 that we tried were all positive, and the 3 that weren't infected were still negative. But if we waited 2 weeks, it was a different story. We only saw 1 of our 3 infected coming up positive. So this is some really promising results that say that people could set up traps, leave them a week, collect them, and then test to see whether they've got Wolbachia and what proportion of mosquitoes are infected with Wolbachia, and be fairly, obviously we need to do more testing, but they'd be fairly happy with the reliability, assuming that this promising result continues. All up, we've tested 79 different sets of mosquitoes in different experiments, and our sensitivity and specificity has been above 95%, so it's really nice looking data. There's also an interesting problem where in the control areas where they spray mosquitoes and try to get rid of dengue, we have emerging insecticide resistance. The way that they check how much insecticide resistance we have in a mosquito is actually by growing them up and then taking a bottle of them and putting in a little piece of paper with insecticide in it and seeing how many die. And the ones that didn't die were resistant to the insecticide. And that's not a very easy thing for communities to do, because you have to know how to grow mosquitoes. And apparently, I was talking to some researchers the other day who work in this area, and they were saying that people have been trying to develop a rapid test for insecticide resistant for the last 20 years without success. And one of the problems is that insecticide resistance can be caused by a single nucleotide change, which is really, really hard to pick up in molecular genetic tests. So for instance, the mutant might have a TGC as part of its genome, whereas the wild type might have a TTC. And so you want a test that will detect the mutant, but not the wild type. So we set out to see if, or should I say Mediha set out to see if she could develop one of our rapid tests that could do this. And her results have been really promising. So this is unpublished. It's fresh off the press. It hasn't been presented before. But what we can see is if we take our mutant mosquitoes, particularly at high concentrations of DNA, we can detect the mutation. But if we take wild-type mosquitoes that have got the same concentration of DNA at this 10 to the 7 concentration of DNA, copies per microliter, we just get a really, really faint band. But then as we dilute down, we don't see it positive anymore. So there's still a little bit of work to be done. And this is actually, when we initially started making them, the first test didn't work at all. And we had to revise and revise and revise. And I'll let Mediha tell her beautiful story. But what we found is that we can adapt our test to detect single nucleotide polymorphisms. A little bit more work might be able to push this over the edge. But we think that there could be some promising, even as it is, if we can work out a way to quickly determine how much DNA you might expect to get from a mosquito, and therefore how much should be added from that extraction step into the assay. We should be able to zero in on a really good protocol that would allow people to very quickly, so we're talking about half an hour test, detect insecticide resistance in their mosquitoes compared to having to grow them up and put mosquitoes in a bottle. So it's really exciting. So if we think about the future work that we want to do, we'd love to combine all these tests together so communities could look for which dengue they have. Do they have the Wolbachia? Is it spreading? Have they got insecticide resistance? Is it spreading? All in a single platform. And we'd really love to move to some on-site trials, and we've been starting to talk to a couple of groups about how this could be done. It's really interesting because when you think about field testing, you imagine standing in a paddock somewhere and you wonder how on earth you could actually do this 3-step manual test in the middle of a paddock. But the reality is that when the mosquitoes get collected, they are always brought back to some kind of facility where they're sorted into their different types, because they want to know how many Aedes aegypti mosquitoes versus how many others. And so it might be a rudimentary room with a trestle table. Well, we know that our tests could be done in that type of rudimentary room with the trestle table. Just, you know, a few quick little steps and you can get a result. So we'd like to start working with some local councils here on the Sunshine Coast, as well as overseas, to see if we can implement some of this. So it's got excellent field potential because it doesn't require power supplies or centrifuges. It just needs a little battery powered heating block and, you know, some liquid handling. And it's highly relevant for testing other diseases other than the ones that we've been talking about. So just as an acknowledgement, Mediha has done some really awesome work. Her supervisors, Nina, Peter, and myself. Leon, Hugo, and Greg Devine have been sharing their infected mosquitoes with us. And Greg was the one that suggested we move towards insecticide resistance testing, so we thank him for the idea. And Angie Vandenherk has also been working with us to talk to us about what level of sensitivity should be required, and sharing his dengue-infected mosquitoes as well. Thank you very much.
Thanks, Joanne, for a great talk. Does anybody have any questions for Joanne? I have a quick question while people are thinking. With the resistance, the classical way they're doing it, they would be identifying, which is, I appreciate, very cumbersome, identifying resistance that's caused by any mutation. I presume that the resistance that you are doing would be only identifying that specific SNP.
Yeah.
So if the resistance is caused by something else, I guess you'll just have to develop a different assay.
Yes, yes. So we can now that we've shown the proof of concept for one SNP, we can then extend to some of the other major SNPs. But one of the researchers we were talking to a couple of weeks ago was saying that that SNP that Mediha chose is one of the most dominant ones in a field trial they're currently doing in Mexico, and that they would be quite eager to try the test there because it's just so, it sounds like it would be so much easier to do than the mosquito rearing, and you'd be looking for the most dominant SNP anyway. So no, it's really, really interesting. So we look forward to when Medica gets back from her maternity leave so she can keep working on the project.
So has she gone already? But she's
Yeah.
going to talk today, I hope, before the baby comes?
Yeah, so she's already on leave. And so I think the baby is imminent. We look forward to hearing when the baby arrives.
Sorry. So Joanne, thank you so much. And we'll move on now to Martina, please.
Yeah, thank you, Abigail. So is everybody seeing the share screen?
Yes.
Yes, excellent. Okay, well, I could just sit here and tell you so much about chlamydia, how amazing they are. But then there was this amazing review, very recent review. It summarizes everything that we want to tell you. There is a chlamydia for every eukaryote on every possible continent. So as you can see, chlamydia have such a broad host range, literally from protists such as amoeba all the way down to very large mammals. So here in Australia, perhaps the most infamous is Chlamydia pecorum that we relentlessly work on because that's one of the most significant pathogens of koala, but also livestock. Of course, we cannot not tell you about Chlamydia trachomatis, which is probably #1 sexually transmitted infection in Australia, but also still major cause of trachoma and particularly important for indigenous populations. And as you heard before lunch from our Susie, well, there is this horse chlamydia. Well, it's not just a horse chlamydia, so chlamydia psittaci infects actually wide range of animals. So I work on this veterinary chlamydia. So I will leave, for example, David in later sections, he's going to tell you a bit more about the human chlamydia, but we think that we did quite a bit of a progress in this animal chlamydia. However, did we? So it's, you know, the overarching question. So we know from overseas studies that chlamydia are gaining diversity, are gaining new hosts, spilling over into new hosts, so they're just being fabulous. And then when we actually looked at here in Oz, in particular for veterinary chlamydia, we are probably most focused on Chlamydia pecorum, very important because it will decimate our cute and cuddly koala. However, we do believe that diversity is perhaps greater than what we are looking for. So we do know, particularly in Australia, we do have a very active human wildlife domesticated animals interface. And of course, chlamydia they are armed and they can be not dangerous, but they can be very dangerous because they have zoonotic potential. They can spill over very easily to humans and cause a disease. So like Susie mentioned in the talks, the vets who were handling the horse infected membranes, they get chlamydia psittaci pneumonia and they ended up in hospital. They had to take antibiotics, so a little bit serious. But then equally, probably one of the biggest concerns is a spill over to wildlife and how these infections affect the wildlife. And this is now cemented by the example of koalas. And then further, we also have an interesting interface, which is so-called of a spill back. Do we have wildlife perhaps spilling back into our domesticated animals these infections? Maybe, as such, we hypothesize for the horse chlamydia, so came from wildlife birds into the horse, the economically significant animal. But all over Australia, so not just us, but other colleagues as well, you know, we all set out on a bit of a quest, and we set out on a quest to find and to seek. So as of right now, so we are screening thousands of samples, and literally we just got new emails during these webinars. Hey, I'm sending you more samples, and we are like, please do. If you see chlamydia, suspect of chlamydia, know of any chlamydia, send us, because we would like to decipher who is transmitting to who this chlamydia, where, how, when, why. So how we do this? So we employ molecular approach. So we screen our samples for preliminary identification and characterization of the infecting chlamydia by using conserved marker 16S, and then we go into focus where we apply whole genome sequencing to even deeper dissect these strains. So I do have to say, we were a little bit inspired by our colleagues from Victoria, who did amazing preliminary work on chlamydia in birds and shared some very important observations. So here is Vasily, and this is actually his quest. So our brilliant PhD student Vasily, at the moment, he is working with a lot of samples from more than 400 birds. And as you can see, he has a lot of DNA to process and to screen. And we were sampling birds together with Australia Zoo. And actually, you will see at the end, we are actually a team Kleiss. So we work together with Renfu, as well as Gabriel Conroy, all born and bred GeneCology. So we look at not just disease, we also look at parasite burden, and we look at conservation of these birds. So all in all, by doing so, was already gave us some very unexpected results, and we were a little bit shocked, just like this stony frog mouth. So okay, let's see what we have found. So we found some maybe good bacteria, not such a scary chlamydia, but we did find some scary one. Well, interestingly enough, chlamydial DNA was detected in many samples. As the screening is ongoing, so we can't give you an exact percentages yet. We have more than 1,000 samples to go through, but our preliminary sequencing determined these novel taxa, which are at the moment of unknown pathogenic potential, and not just of the pathogenic potential, but also of the zoonotic potential. But then again, we saw chlamydia pecorum in some of the cockatiels. That's the same chlamydia that infect your koala. And interestingly enough, it was the cockatoo that we saw chlamydia pecorum. And then, if you do recall from Susie's talk in the first session, we have also detected chlamydia psittaci, as well as related chlamydia abortus strains. These guys have the zoonotic potential to humans, as well as they cause pretty severe events in horses. So what is the actual spillover risk? How are they dangerous to us? So there are more questions than we actually can give you the answers. And then we thought, well, are just the birds to blame for these spillovers? And we can immediately tell you, because we also look at other hosts, livestock, other marsupials, definitely not. The epidemiology of these infections, it's actually pretty complex. We think we have some ideas, but I bet that all of our ideas are gonna go away. So now that we have detected chlamydia in birds, are the birds now some kind of a reservoir for koala? Then further, are now the birds impacting other livestock than the horse, etc.? So what we do is, so we decided, okay, guys, well, buckle up. We got to continue on our quest. And we can tell you that on our quest, and further, we have found even more stuff. So for example, we do know that several species, there is a belief that they are exotic and that they are not found in Australia. However, we did found chlamydia abortus in birds. Is it related? We don't know, so we need to dig a bit deeper. Then again, there is always a question about cats, dogs, our pets. Do they carry chlamydia? Well, yes, we actually did found some chlamydia in cats. That has been known for a while, but it's not very common. But it is important that we alert the owners, because these chlamydia are also zoonotic, and they are documented to give zoonotic disease. Then again, this novel chlamydia in birds, our colleagues from Deakin University, they did amazing work, and they describe this emerging species, not just in chickens, affecting economically important chicken, but also galah. And again, galah being wildlife, are we spilling back? Are we spilling over? So who knows what is happening? And then again, we also have other marsupials. We have wild deer, wild boar. So we can tell you we have found some of the novel taxa in deer, etc. And then, interestingly enough, while we couldn't find any reports for chlamydia in wombats and or kangaroos, we actually found chlamydia psittaci, the same one, like the horse, like the birds, like the humans, in wallaby. So all in all, we are on a quest. We do hope you can join us in the quest. And of course, all this research, it's actually not possible to do if we didn't have so many partners. And in here, I can't even name all the partners and all the participating vets and colleagues. And if you can see, so this is our lovely and charming chlamydians. We'll have a couple of incoming, so stay for their talk. We thank Peter Timms, we thank Abigail for always supporting us. This is MJ Lab, and of course, Team Lice, Chlamydia and Lice with Renfu and Alan. Thank you.
Thank you very much, Martina. This is indeed scary. And I have to ask, what about dogs?
Well, in Europe, interestingly enough, the reports from Europe, they did identify chlamydia in dog polyarthritis. So working with IDEX, we are now also arranging to look at the dogs. So we're going to assess actually joint fluids. That's incoming tomorrow. So I will keep you guys posted. But all in all, as you can see from my second slide, we have so much evidence that chlamydia is perhaps your ultimate ubiquitous animal. So in humans, chlamydia can cause polyarthritis, so in livestock as well. So there is a plausibility that it can cause that in dogs, for example.
Okay, I'm still sleeping with mine.
Me too. That's okay, we share a microbiome. Perfect.
Any other questions for, oh, there's a question from Ajit. What is the mode of transmission to humans?
Well, Ajit, we believe that it's via inhalation, via aerosol. And then as Susie elucidated before, so there are a couple of barriers, you know, for us that we get wildlife chlamydia, zoonotic chlamydia. That chlamydia has to be in high concentration. So for example, if you see flock of galahs or cockatiels, maybe kind of look at them, you know, from the side. Don't go there and inhale. So we do believe that it is via inhalation.
And there's a question from Samuel, are there common tick species that carry different types of chlamydia?
That was the research from our previous researcher, Dr. Delaney Bernard. So Delaney investigated some of the ticks for chlamydia, and we believe that we have found this so-called environmental novel chlamydia of unknown pathogenic potential.
Okay. Well, obviously still a lot of work and really important work. Thank you,
Yes.
Martina. It's been wonderful.
Thank you.
And I'd like to move to Josephine now, please.
Thank you, Abigail. I'll just share my slide now. Okay, can everybody see my slide?
Yes, thank you.
Okay, thank you. I'm presenting my